So I see everybody is back for this first session of Functional Metagenomics 2022. My name is Alexander Wenzel, senior researcher at Center for Industry in Trondheim, Norway. And I really would like to thank Trevor and the whole team at the University of Waterloo for setting up this nice conference and also enabling it to be streamed live and recorded. I'm happy to chair this first session, which is called Sequence Facilitated Functional Metagenomics. And in that session, we will have 3 lectures in total. It will be 3 plenary lectures and 1 selected on posters. And yeah, without any further comments, I think I will just hand over the word to our first speaker, which will be Intikab Alam from the Computational Bioscience Research Center at KAUST in Saudi Arabia. And his lecture will be titled 1 Billion Genes Catalog and Metagenomic Assembled Genomes. Intikab, the floor is yours.
Thank you, Alex, for introduction. Can you guys hear me? Is my voice clear or?
Yes, we can hear.
All right, thank you. First of all, thank you so much for organizing this conference. Previously, I attended the one in Norway in 2019, and it was a wonderful experience. Looking at the work coming from different aspects of metagenomics and functional genomics. This time I wanted to come, but there were some reasons I couldn't travel. But I would still like to contribute to this conference through sharing our work. Some of this was previously presented in the previous conference. So first, I just give an overview of my presentation. I'll just give a brief introduction to metagenomics, some of the issues and opportunities. Then I will give some background towards KAUST metagenomic analysis platform and some of the data sets which we have published. And then near the end, I will talk about the world's largest gene catalog and related metagenomic assembled genomes with some outlook of functional metagenomics. So in general, we all know metagenomics is direct sequencing from samples which can be taken from environment or a host associated samples. And there are 2 aspects of this analysis. One is direct functional driven analysis. The other one is sequence driven analysis. And mostly this talk would be about this computational part of the analysis, where we want to know who is out there and what are they doing. So just to give you a brief background, the first reports of what you call observations about oral microbiota were reported from 1676. And then another report from 1888 shows Robert Koch isolates microorganisms using solid cultures. And this continued till the discovery of the DNA. And then the PCR was made. And then Dr. Handelsman, we are honored to have her give the first lecture as well. And she introduced the first use of the term metagenomics. And after that, with the development of next generation sequencing methods and machines, the number of studies started to grow. And by 2015, we had around more than 1,000 studies done already. And what we see since then is that there's a shift of bottleneck from data generation to data analysis. And the last big study we have seen is from 2015 from Tara Oceans. And before that, like human gut gene catalog. Human gut gene catalog showed around 10 million unique genes. And from Tara Oceans, they showed around 40 million unique genes. And this shift of bottleneck from data generation to data analysis required some fast methods and what you call it becomes a bottleneck in the computational requirements but the main goal is to reduce the number of experiments at the end to do more specific targeted functional metagenomics. So in general, what we see is, why is metagenomics useful? We try to look at the taxonomy and function. We try to see what we call in the dark matter. It becomes the gold mine for novel genes, which has many research and applications in health, biotechnology, novel enzymes, and many others. And what we see is large number of samples are available from different environments already. And these large number of samples which are available, what we see over time, the methods or techniques with which these were analyzed were changing over time. And the sequencing technology used was also changing. And the methods which were used to analyze this data, some were directly based on reads, which were shorter, and some were based on assembly-based analysis where the shorter context could be obtained. But what are the issues related to it? So if we work on this kind of data, there's a possibility we get partial genes, and annotations could be very limited, and data integration becomes a problem when the data is analyzed over time using different methodology and techniques. And this creates a big bottleneck that how we compare any new data with the previous one. And looking at shorter genes, what we will have as a result, that means partial in, partial out. So it becomes more complex later on to do functional studies. So if we look at these three main issues, are there any solutions? For example, there are some methods which can stop predicting the genes near the ends of the context. So all the genes before that can be forced to be predicted in a complete form where start and end is detected. And if we solve this problem, then the other problem is the existing older samples were annotated with the older databases and older methods, and then annotations are quite limited. So as the reference data sets grow, it requires, that means we could do re-annotation and get some more better results. And for data integration, there's a possibility to create a simple user annotation table, which I will show in the coming slides. And this could result into an analysis which is based on complete genes. And this will not be affected by changing the sequencing technology, for example, the long reads or short reads. So complete genes will always remain complete genes. And these can be useful to look at in a functional point of view. So when we try to look at potential solutions for this, and we look at large number of samples, so we proposed and published now this work on KAUST metagenomic analysis platform, where we give a concept of improved metagenomics, that if we have samples from different studies, and if we try to assemble them using a standard technique for all the samples, so this will result in slightly larger context with a possibility to get the complete genes. And if these complete genes, we try to do clustering, as we do in step 2, for example, and this clustering will yield in a table where, say, columns are genes and rows are samples. And this matrix can be used as a standard. This is the full set of genes, non-redundant set of genes, which appeared from a particular gene catalog from a particular environment, for example, or as a whole. And these complete genes could then be annotated with the latest reference databases, trying to understand for each gene, what is the potential function and what is the potential taxonomy. And this could be then used further for mining or comparing different samples. This creates a big bottleneck otherwise if you look at individual samples, but this technique, I think, makes it easier to look at this matrix and try to see what different samples do have in common and what are unique set of genes to different samples, for example. And using big computational resources, like we have a system at KAUST, which is called Shaheen, is a supercomputer. Combined with our method, we analyzed existing gene catalogs and created some environmental data sets-based gene catalogs, which I'll show in the coming slide. And this could be then looked at for further analysis. And this particular procedure requires There's some memory intensive steps, for example, some compute intensive steps where you try to compare your complete gene with the public reference databases. And then at the end, a mechanism in which you could either computationally on the command line analyze the data, or you have an interface, web interface for users who are not expert in bioinformatics. They could still analyze the data, trying to find similarities and differences in different data sets. And one aspect which I mentioned previously is that we could use a common simple annotation table. For example, here I'm showing the columns of annotation which are shown as rows for better visibility, that you could have different features of a gene as much as possible, including the scores with which you find a match in public data sets, so that later on, if you want to analyze that set, you can filter it according to your criteria. You can search it based on identifiers from some reference data sets from, for example, KEGG or PFAM or gene ontology, or for a particular taxonomy, and it can yield the results in a way which becomes easier to look at. In all data sets, if they have this format, then it's easier to analyze. So there is a system produced by Craig Venter Institute previously. It's called MetaRep, which could be adopted for using this kind of table to either browse, view the results, or even to look into deeper into taxonomic classification, functional classification, or compare the data. And this can be adopted by any group, I think, when the data has become available in this kind of table. And we introduced additionally some filters to the data that, for example, from the whole gene catalog, you are only interested in a set where KOs or from KEGG-based KEGG ortholog data is available, or a particular enzyme set is available, or some other filters which are available. And to perform all this annotation, we developed this Chaos metagenomic analysis platform, where you can start with either a genome or a metagenome context, for example, or you can use CDSs, or if you have protein sets from even eukaryotes, can be put into the system. There if it's protein set, for example, we go for a direct blast against KEGG database and UniProt full database, trying to extract the information for the closure function and potential taxonomy. And alongside, we do InterPro, where we can look at what potential non-protein families are present in the gene which we are looking at from PFAM, ProCyte, and TigerFAM, and others. And then as a result, we get a table which is called a simple annotation TSV table, which has 19 different types of annotation included. And we also get results in GFF or FASTA files. And this can be then used for visualization of hierarchies from functional databases like KEGG or COGS or gene ontology or InterPro, or a particular dictionary which we made here. We call it industrial POIs, or proteins of industrial interest. I'll talk about this later. And then this can also be used for visualization through MetaRep, or it can also show some individual level graphs. But for comparisons, you can use this kind of table in MetaRep for viewing or comparison. And if you look at existing features in KMAP compared to other pipelines, for example, MGRAST or EBI metagenomics or MetaPipe, or in itself MetaRep or IMGM, what we see is there are many features which are available in these pipelines, and but there are some which are missing in one or the other. In our case, for example, we don't directly work with reads, so that part is missing, which is shown in orange. And what you call it, this system is available online, so it can be used for local installation, but it can analyze data for several different aspects, which are some of those are available in other pipelines as well. But overall, what we show here is that KMAP has some more capability compared to many different pipelines. For example, EBI does annotation of function based on PFAM only. So we don't know this particular gene, which do have a particular PFAM family assigned, what could be the potential taxonomic origin of this gene. And if it's not there, it becomes difficult later on to try to understand the potential relationship with taxonomies. So in this system, what we published recently, there were two pilot projects. One was where we tried to look at existing gene catalog and try to re-annotate those and see whether we improve upon the unassigned function of portion of the gene catalog, which are there, for example, from Tara or human gut microbiome or Tara eukaryotic gene catalog. And second part was that we selected randomly around 36 different habitats based on the metagenomic types which were available from European Nucleotide Archive, so around more than 1,000 samples. And then additionally, in addition to annotation or before annotation, we had to do some pre-processing. For example, the metagenomic assembly, full-length gene prediction, and then producing a gene catalog, which yielded around 275 million genes across these environments. And this is available for looking at this data it's all data is available publicly. In addition to this, because we started to look at around 27,000 samples obtained from different environments from European Nucleotide Archive, and all these assemblies were released with this publication. So this is a website of KMAP where you can go and then you can look at what existing annotations are available, and then all these 27,000 assemblies are available for users who want to look at through their own system. Now, coming to the existing gene catalogs, for example, Tara and human gut gene catalog. So when we look at the portion in terms of taxonomic assignments, what we have seen is that the first version had around 20 million genes in human gut gene catalog, which were assigned certain taxonomy because at the time they looked at the comparison with the complete genes only. So in our case, we were able to increase it to, additionally to a larger number, which is 79, around 7 million, for example, so around 8 million genes could be assigned with taxonomy. And similarly for Tara data, 23 million genes could have been assigned in the original catalog. In another case, we extended it to around 28 million genes for bacteria. And similarly, the improvement was seen for other assignments. Additionally, we did re-annotation of 2 more gene catalogs where we see some improvements. And here what we show is that the original gene catalog had, from Tara Ocean, had around 30% of the data which was unassigned. In another case, we reduced it to 21% when we have the coverage of genes up to 80%. And if we reduce it a bit, it goes to 18%. And similarly for human gene gut, for example, we had 78% of the genes which had no taxonomic affiliation. So we reduced it to 22% with the conservative coverage, and then some loose coverage, we can go it to around 18%. So it's a big improvement. So what it means is, because public data sets, reference data sets are improving, so it requires re-annotation at some stage to improve the understanding of the data. Similarly, on function, we were able to reduce the unassigned person for Tara from 28% to 16%, and for human gut gene catalog from 35% to 16%. So it's also a big improvement. And then the second pilot project which has been published with the study, we collected 36 different habitats based on the names of the metagenomes, which are from different environments, from water, soil, host-associated extreme environments, and some engineered ones. And we performed metagenomic assembly, full-length gene prediction, gene catalogs for each habitat, which yielded around 275 million genes. And then we used the online processing module for annotation, which at the end yielded 36 gene catalogs. So this is just an overview. That means the standard system is doing only annotation. So if we had to feed some data from metagenomes, we had to do the assembly separately in offline pre-processing, and similarly the gene catalog production. So once we have gene catalogs produced, these complete genes were then submitted to the KMAP annotation pipeline, which then yields the results in a a simple table format, which can then be used to produce visualizations through metadata. So if you look at the size of the gene catalog, the number of genes, what you see is it's nearly what you call a 50 million in Tara eukaryotic gene catalog. In soil, for example, nearly a second to this one, and the number grows on. So this is around, I think, 275 million genes. And if we had only one core computer, it would require really around 500 years to compute a blast against unique plot folder cells. So it's which is not possible, I think, which was not possible previously. But now, given the computational resource which we have at CAUS, we were able to do this in around 13 days using 4.8 million core hours per day. So this computational resource and intelligent methods are able to provide answers to these difficult questions. So for these 36 gene catalogs, what do we see? So if we start from lower side of the picture, what we are showing is to the left, these are, say, representative genes or the from gene clusters. So it shows you in numbers what is the size of gene catalog, and these black dots are showing what you call it, how many gene clusters are there in millions. And then the red or orange dot shows how much in percent is annotated, and percent is shown on the right side. So what we could see is we could annotate quite a lot through this particular analysis, although some of the gene catalog from soil, for example, or eukaryotic gene catalog, the annotation percent is not that high, but overall we can go 60% and above for each of these gene catalogs. So in the middle part, I'm showing the proxy of 16S kind of analysis, taking a single copy gene, RPSC, and what we are seeing is the proportion of each gene catalog in numbers where we see archaea, bacteria, and eukaryotes, microeukaryotes. And the top part of the picture shows for these representative gene clusters, what is the proportion of enzymatic classification from different classes of enzymes. One additional feature which I mentioned earlier is the proteins of interest or proteins of industrial interest. And here we made a dictionary based on looking at the literature and many different databases, trying to figure it out what enzymes are useful for different industries. And then we made two categories. One at the top level, for example, say agriculture, and then within agriculture, what kind of enzymes or genes are useful in animal feedstock, crop protection, in analytics, for example, biosensor or diagnostics, and similarly for energy or many other industrial categories. And this assignment is done automatically when we analyze a genome or a metagenome through our pipeline. So we know this group of genes or enzymes can be useful for this particular industry. So it's a good way of looking at the potential applications. So one example is here, looking at some cellulases, alpha amylases, or catalases from different environments. It shows different proportions which are available from different gene catalogs. And all this data can be mined using the online interface of KMET system. So overall, what I've shown so far is a published work where we showed large number of samples are available. And if we are able to reanalyze this data from scratch, trying to assemble it to a reasonable size, and from there we predict complete genes, make gene catalogs, annotate the gene catalogs, produce the data in a nice and simple table, which can be integrated into any other pipeline for visualization or comparison, and try to look at the data either computationally or through these interfaces, which can take these tables and try to help the non-expert users to look into different features of the data. So now this gives us a basis to look at those 27,000 samples which we assembled, and we only published a portion of it because at the time it was not fully annotated due to computational requirements. And for this 27,000 samples, now we have complete metagenomic assemblies available. And this produced a gene set of non-redundant genes, around 3 billion genes. And when we cluster all this, it gives us around 1 billion unique genes across these environments and samples. And then all the annotation for this 1 billion gene was processed through our KMap annotation system. So on the left side, I'm showing the pre-processing, which was done before we started the annotation, which requires assembling, gene clustering to make the gene catalog, the world's biggest gene catalog, and then submitting it to for annotation to our annotation system, KMap. So here, what I show you is the number of samples from different metagenome types, which are available from European Nucleotide Archive. So at the top, you see human gut metagenome, then skin and marine metagenome, soil. And there are some which just says metagenome, for example. So it's hard to understand from the existing ontology or the way the data was organized over time that this metagenome is coming from which particular type of environment. And just before I explain the data, I mean, all these assemblies which can be available through this online interface. This is an example showing how you can retrieve the data. For each metagenome type, you will see in the beginning the name. When you click on it, it shows you what projects are there or the project IDs or study IDs from ENA. And then the state statistics on the samples, which is shown in the last portion of the slide. And then in there, you can see the assembly statistics for each of the sample and alongside a link to the metadata so that we can query the information around the samples. So if you look at these 450 different studies, these are coming from, the largest number comes from a section called metagenome. So we don't know originally based on just looking at these types from where this data is coming from. And similarly, other sections do have right ontology like human gut metagenome and others, but there are some where it was not clear. So what we did is, here we are showing the number of samples, and alongside what we did is we looked at the metadata for several individual samples and trying to figure it out what could be the potential source, type of the biome or habitat from where this data is coming from. And alongside, we had made a decision to use the information with each, which is available for each sample. For example, the project ID from ENA and the sample or run ID from ENA, so that even the gene names contain this information. So if we had to trace back to the original samples to locate the metadata, it becomes easy. So alongside, we looked at the ontology of existing samples, looking into potential publications and other metadata which was available for individual samples. Then we were able to make these four main categories: ocean, host, associated, land, and engineered, which are then sub-classified into sub-habitats, for example, or biomes. And then here I'm showing to the left the numbers, which are the numbers of gene catalogs or unique genes, and these are in millions. And on the right side, what you see in bracket, these are the number of samples. For example, from pelagic, we have around 2,000 samples, and we had 298 million unique genes. And from host associated, we see 17,000 samples from human, around 5,000 samples from other animals, and the number of unique genes are about 151 million from human and around 136 million from animals. So this overall constitutes a gene catalog of about 928 million gene clusters. And if we try to make a subset of it, then what portion of this is annotated? So this makes around 60% of the gene catalog, which is annotated. So the rest of the slide will show the data from annotated part of the gene catalog. So here I'm showing the same data, but the middle figure is showing what proportion of taxonomic assignment could be done on this data. So you could see first the bacteria, archaea, archaea, and then viruses. So this is a proportion from different samples or these biomes, which can be looked at for the taxonomic breakdown. And on the right-hand side, we show the potential functional grouping of this data. For example, it shows around 80 million genes, which are shown from genetic information processing. And if we go on, we included a section on antimicrobial resistance genes. So it seems there are about 2 million unique genes in the whole data which we have, which would be interesting to look at. And if we look at the sharing among different biomes and subbiomes, what we see is more genes are shared within that particular biome than shared to others, which is also confirmed by another gene catalog, which was recently published by Pierbock Group, where they found around 302 million gene clusters. And they also concluded that between these different biomes, there is less sharing. More genes are unique to a particular environment apart from the human related data. So now we are developing a database of all this data to look into some more detail. And if I take from one example of, say, a PETase enzyme, trying to see how many hits we get from different environments, it shows around 5,000 hits from several different environments, and these genes could be looked at for functional analysis in future. Similarly, if I look at the proxy of the 16S gene, which is RPSC, so what we see is around 129,000 different unique genes pointing towards different taxa. So it's a huge number of taxa which can be covered based on this particular annotation. And similarly, when we look at the POIs part, we have around 128 million unique POIs which are related to different industries. And all this data is available through the database, but still it's in development, not shared outside. And the same data, the 27,000 assemblies, when we look at trying to bin the data for building the metagenomic assembled genomes, we have obtained around 600,000 megs. But if we do the dereplication, that we don't have any two megs repeated twice, then we have around 120,000 metagenomic assembled genomes. And out of this, there are 60,000 which do have biosynthetic gene clusters. And what I'm showing in the figure, the last part shows NRPS and PKS related mags. So in total, we have around 15,000 mags which do have biosynthetic gene clusters. And then now is the time that this data could be then analyzed, functional aspects of this data. And for this, what we are looking at, there are around 400 million genes which did not get any head to public databases. So there it requires structural-based inference and then trying to see what potential functions are there. And for the other part, about 128 million genes, we can see based on some model enzyme that what are the differences, can we find some better enzymes, and trying to do some more functional analysis. So here I will conclude my talk, and then I will just acknowledge the other colleagues who are part of this work. Professor Vladimir Bayek, who is now no more with us. He died in 2019, but he was with us from 2009, making this Center for Bioinformatics in KAUST. And then all of the group, without their contribution, this would not have been possible to reach at this stage. So I end my talk here. If you have any questions, I would be glad to answer.
Thank you very much, Intikap, for this impressive work and trying to structure the vast amount of data that is out there in public databases and try to provide some lines for further research in the enzyme discovery world. Is there any questions from the audience? There's one from Jang-Son from Center.
Hi, yes, I'm Yang Sheng from Synprep Industry. So,
Hi.
Thank you for the nice talk. I have some questions.
Yes, please go ahead.
Yeah, what do you think about the usefulness or the role of structural prediction for annotation? Do you think that it will really help with the annotation of very novel enzymes? Is the first question.
It's a very good question. And I think if I could answer this one, then I can have the second question. So, I mean, this is an area which is now made slightly easy by the development of this AlphaFold, with which I think you can generate models of structure close to potentially real structures. And that could be a very good start, but this would require some sort of ontology, which could be based on experimentally produced structures where, say, for different folds, if there is a possibility of a relationship with certain type of functions, for example, for each unknown gene, if you're able to make a structure and it shows different folds and other structural information, and that taken from the annotations from known structures, if you're able to assign 2 or 3 functional types, so it could be possible to do 2, 3, or 4 different functional tests to see, do we see some result? And that might be a way to go forward. But when you have large number of data sets, it's not that straightforward, I think. Second question?
Yes. Do you think there's a bias of the samples that you analyzed?
Definitely there is, I think, because what you see is that, what has been over sequenced and what has been under sequenced till now. So what we see is human related sequencing is much higher in numbers than compared to other environments. Although there are some other projects starting, for example, Earth Microbiome Project and others, where they try to see if they can increase the sequencing coverage for some of the other environments, and that will really help. But we try to look at whatever is possible. And then what we have seen is there are additional samples. We analyze the data till what was submitted to ENA till 2019. So I see there are additional 20,000 samples, which could be looked at, and then it will include, I think, some more information. So we take it from there. But I think this field is now recognized that metagenomics can yield a lot of information about different environments. And these big projects like Earth Microbiome, they started to sequence these under-sequenced habitats, for example. So it will be a good addition in the future.
My last question about the POI classification. So because some enzymes have many different applications, so how do you classify them?
So these are then classified into multiple categories, because we know that there's some enzyme can be used in different industries. But then this is now shown if you have a set of interesting maybe data sets or a genome or a set of proteins, you can pass it through our annotation pipeline, which is available online. And you will see in the results, that means it will show you that which genes seems to have multiple applications and which genes are there, which is unique to certain applications. So this assignment is done for whatever data you try to put to our system.
Okay, we have time for one more question, if anyone has from the audience. Yes, please.
Hello. Yeah, thank you, Professor Alam, for that talk. It was very inspiring. And as somebody who's done a lot of hair pulling when I'm trying to put together data from all different kinds of platforms and annotations, this really seems like a one-stop shop. But just to build on that, I wanted to ask, so I saw that the assembly and there are a few steps that are done offline, you said. So
Yes.
if somebody does, like, how agnostic is this platform to handling like assemblies that come from different types of pipelines? Because
Yes.
we know that there are quite a few differences. And also, just generally speaking, in each step, like how agnostic it is that we can get an input from some other tool or pipeline and then feed it into this metagenome annotation pipeline and still get what we want. Yeah.
The answer is in your question already, because for that particular reason, we did not include the assembly mechanism to be included in the pipeline, because technologies with which the data is being sequenced is changing. The sequencing depth issues, for example, long reads can give you longer context, but then issues with coverage. So what we provide is, if you go to our website, it shows you the methods with which we did the assembly. at least the data which we collected is we limited to Illumina data sets, reasons of high coverage so that you can have better assembly, and then the paired end data. That was the source of the data which we tried to assemble. And if you, there are many assembly tools available for different types of sequencing platforms, I think. So people could try, because it's becomes on the individual taste how you want to combine this. Sometimes people have dual sequencing done, one with the long reads, one with the short reads, so that they have coverage and the long reads as well, so that they can recover almost full genomes from even metagenomes. So in that particular case, there is quite a bit of variability. For this reason, this assembly option is not available online, but we show methods how we did it through the paper you can see, or the website. And this is based on Illumina. And if any other what you call a long read technologies are used, there's some specific methods available, and those could be used. So even eukaryotic data could be put into our pipeline, but at the protein level then. So you can submit proteins, and then it will analyze all for you. So due to complexity in requirements for assembly in different types of sequencing, we did not yet include a module for assembly, but in future, we may be able to.
Okay, thank you very much, Intikap, once again.
Thank you.
Yes, and then we move on to our second and first in-person talk here today, which will be by Camilla Nesper from the University of Toronto, Canada. And the title of her lecture will be Bathyarchaeota archaeota in anaerobic digesters treating pulp mill wastewaters. Camilla.
Okay. Thank you. You can all hear me in the back. Good. Yeah. So before I, if I can move forward, would be good. I'm pressing the green button. Okay. Oh, there we are. Excellent. Okay, yeah. So this is a part of a larger Genome Canada sponsored project. And Anupama, Sharon, who's here, and she's also going to present data from this project. So the overall goal, it's the project is called Symbiomics. Overall goal is to investigate biocatalysts that kind of upgrade the wood part of pulp mill, what comes out of pulp mill, so cellulose, hemicellulose, and lignin. Okay. Oh. I should not have talked so much because I'm pressing it again. Yeah. Okay, you do it. Okay, yeah. So yeah. So before I start talking about these anaerobic digesters, I thought I should give a little bit of background into who these Bathyarchaeota are, since not everybody might be so familiar with them. So they used to be called the miscellaneous equinocardial group. They are widespread and can be very abundant in anoxic environments. They are very common in deep subsurface environments, which is where they got their name from. And since the deep subsurface is probably the largest biosphere on Earth, they are also among the most abundant cells on our planet. But there's still no cultivated representative of these organisms. They're only known from 16S RNA studies and from metagenomes, where people make metagenome assembled genomes, which is what we have been doing too. There's been several papers out recently kind of investigating where we can find them and what they're doing. And this is a great review. And I took this 16S RNA gene tree from this paper. So here they show that there are 25 subgroups of these Bathyarchaeota. The most abundant ones come from the pink, which is called MCG8, and the red, which they call MCG6. And these are also the groups where we see our MAGs come from. So next slide, please. Okay, so these are just some of these studies that have looked at what these organisms do. When you look at most of the MAGs, they have a kind of a full archaeal pathway or version of the Wood-Longdale pathway, suggesting that they are cytogens, that they can grow autotrophically. This paper here was probably one of the first ones that showed that their growth is stimulated by growing them with lignin, suggesting that they use lignin as an energy source. And they suggest that they use methyl or methoxy groups from lignin itself, or from degradation products of lignin. This paper here looked at these Bathyarchaea from termite guts, so they are quite common there as well. And they also suggest that they kind of use lignin and that they use methyl groups from lignin or lignin degradation products. This paper here came out just a few weeks ago, and here they show that they could stimulate growth of these Bathyarchaeota in enrichment cultures on lignin and on this syringe aldehyde, which is a lignin degradation product. And I've just shown a little picture of it here. It's kind of a phenol with an aldehyde group and some methoxy groups. Can I get my new slide? Oh, did I do that? Yeah, haha, it is working. Hi, yay. So yeah. Yeah, so I just wanted to show you these pulp mills at the digesters at the pulp mill. So we have the MAGs from two different data sets. So the first one is from these anaerobic digesters at pulp mills. So I just wanted to show you that these are actually kind of industrial sized bioreactors. It's not like these small ones. This one here is huge. So this here is people standing on top of it. And so we are interested in kind of what are the microbial communities in these digesters, and how do they change over time, and how are they linked to other operational parameters from these digesters. Okay, so Ivy Yang, she is another research associate at Biozone. So is it possible to take this off? So no, okay. Anyway, so she has done he's done a lot of monitoring using 16S RNA sequencing of these digesters. So I think she has about 200 samples that she's looked at, but here she has grouped them into years and seasons. And this here just shows the archaeal community. So you can see there's a lot of these methanogens in these reactors, which is not surprising since they make it's kind of methanogenic bioreactors. But we also see these Bathyarchaea, and we don't know exactly what they are doing. They can be very abundant in these systems and make up to 10% of the archaeal community. Torstein Meyer, he's another research associate at Biozone, and he has looked at the correlations between these ASVs and kind of operational parameters in these bioreactors. So he has found that these bathyarchaeota are among the most sensitive to changes in operational parameters in the mills. He found that they are always positively correlated with hydrogenotrophic and metallotrophic methanogens, and negatively correlated with acetoclastic methanogens, and it's for all the 3 mills. And we don't know if this is because they like the same conditions, or maybe they have some syntrophic relationship with these methanogens. He also found out that one of the mills are linked to color production, and that color is a byproduct of lignin degradation. So that kind of suggests that they might be involved in using lignin in these reactors as well. Okay, so I just wanted to show you like the main steps of this anaerobic digestions that happen in these mills. So first you have this hydrolysis of the complex polymers, and then these are broken down to smaller short chain fatty acids, and then I converted to acetate and hydrogen and CO2 that is used by the methanogens to do methanogenesis. So these 2 first steps is usually attributed to bacteria, so fermented to bacteria and bacterial pseudogens. And then of course the archaea does this last methanogenesis part. So we are interested in where these kind of bathyarchaeota fit in. And to look at that, we did metagenome sequencing. So we sequenced 2 samples from each mill using Illumina, and then we sequenced 1 of the samples using PacBio, and did assembly and binning, and this gave us 16 of these bathyarchaeota mags. So we did a lot of manual refinement to get the 16 assays into the mags, for instance, so we could kind of connect it to the amplicon data. And this resulted in us closing one of our mags. This is the first closed genome of a bathyarchaeota. And it's quite a lot bigger than, so it's about 3 megabases, quite a lot bigger than the mags that are present in GenBank for these, and IMG for these organisms. And overall, all of our mags are larger than what people have submitted to GenBank. So I think that is mainly because we have this PacBio sequencing and we can get longer reads and longer contigs. Okay, so the second data set we looked at is from some cultures that we have. So they were set up by Mabel Wong. She was a grad student at Biozone. So she made 6 cultures on cellulose, so 3 on cellulose and 3 on poplar hydrolysate. And they were incubated with, or inoculated with beaver droppings, moose rumen, or granules from one of the mills that we have been looking at. And she did metagenome sequencing, and then we made these mags again. So we have 19 bathyarchaeota mags from these cultures. So I've shown you, this here just shows the abundance of these mags and the cultures that we have. So we have abundance here, and then we have mag ID here. So I just wanted to show you that we only see them in the poplar hydrolysate cultures. They're very, very low abundance in the cellulose, which is shown in blue, so you can't really see them. You can only see the orange bars. So this suggests that they cannot use cellulose, but that they use something else in the poplar hydrolysate, so which is composed of glucane, lignin, silane, and mannan. So they probably are using one of these substrates as carbon source. Okay, can you put it down a little bit? No, just so we can see the top. Yeah. So this is just a polygenetic tree of our mags. So here we took mags from the genome taxonomy database, and just to see which of the clades they fall into. So the ones in black are from the mills, the ones in gray are from the, no, it's fine, are from the cultures. And anyway, you can see that most of them fall into this MCG6 clade here. the ones in triangles are just from GenBank. And these MCG6 genomes have been found to be common in termite guts, in permafrost active layers, soil, and in other kind of bioreactors. And they fall into these three clades that are just called clade A, B, and C. We also see one mag that falls into this clade that is very closely related to the MCG6 group here. So in my figures, I've only called it 6D. But in the genome taxonomy database, they have this SG8323 is their name. And then we have two mags from this that are more distantly related to the others, and they fall into this MCG8 clade. That first paper that showed linkage to kind of lignin use was from this clade. But that a recent paper where they could enrich them on syringe aldehyde, those patharchote are from this MCG6 clade. So it looks like both of the lineages that we have, is that me? No. Okay. Can be linked to lignin use. No, I can't do it again. So can you take my slide forward, please? Okay. No, back. Okay. No, forward. Okay, I did. No. Okay. So we then did a pan-genome analysis to compare the genomes and look for genes that kind of associated with the different groups. So this here shows our pan-genome plot. So here, each of these rings are a genome, and each of the tiny little lines is a gene from a gene cluster. And so the ones in black are from the mills, and the ones in gray are from the cultures. And you can also see we have some very thin black lines here. Those are genomes that we took from GenBank just to have a larger collection of these MCG8 genomes so that we could do a comparison and see are some genes associated with these MCG8 genomes or in the MCG6 group. I've also included this tree here. This just shows the clustering of these mags based on the presence absence of their genes, and we see kind of the same clustering patterns overall as what we see from the phylogenetic tree based on these conserved genes that I showed in the previous slide. Okay, so this here just shows a heat map of some of those interesting genes that and pathways that we found in these genomes. So the top one just shows that they all have so each of these columns here is a mag. Each of the columns is a mag, and each of the line is an interesting gene or a pathway. And I've made this one in red here because this is our closed genome. So they all have the pathway for cytogenesis, so which is what people have seen before, so they probably are cytogens. They have a lot of genes involved with hydrogen metabolism. The ones in blue indicate that they were overrepresented in the MCG6 group, and the ones in green were associated with MCG8 group. So the MCG6 genomes have genes for methyl metabolism. They have a full vitamin B12 or cobalamin biosynthesis pathway, which is quite interesting. This is 13 genes that they have. They can do nitrogen fixation, and they have more genes for beta oxidation than what we saw in the MCG8 genomes. While these MCG8 genomes have more genes for glycogen degradation. They also have this UBX gene here, which is interesting, and I'll come back to that later, but this could be involved in degradation of aromatic compounds. Some other interesting genes that kind of, they vary, but not between the groups. Some of them have a flagella, they have a ribisco that they probably use to fix CO2 into nucleosides. And then they have this heliorhodopsin, which is a light sensing protein, which is quite interesting, that these subsurface or bioreactor genomes have heliorhodopsin. They probably don't see light very often. There. Okay, so also this another pathway that we saw in these in the closed genome was the full TCA cycle, and I just include that here because it kind of illustrates some of the problems with working with kind of unknown genomes and MAGs that are incomplete. So when we looked at when we did automated annotation of the genomes, they were missing several of these genes in the TCA cycle. But Ilya Kublanov here, he is an expert in metabolism, and when he looked at and did manual annotation of the closed genome, he found that he could find homologs of all the genes in the TCA cycle. So this just shows the putative TCA cycle in the closed genome. So a homolog in the closed genome is indicated by this purple circle here. I've indicated if this found in these other clades by these other colored circles. So if they're found in all the MCG8 genomes, they have a closed circle. If it's found in some, they have like an open circle. So for instance, this isocitrate dehydrogenase was not detected by the automated annotation, but when he looked at it, he could find a homolog in the closed genome, and then we could find homologs in all the other MAGs that we had. Some of the other ones we could just find, we could find a homolog in the closed genome, so this fumarate hydratase, for instance, but just some of the other MAGs have this gene. So this could be due to that they don't have the gene, of course, or the MAG is just because the MAG is incomplete, or maybe they have a more divergent homolog that you need kind of automate or need manual annotation in order to find it. Okay, so now I'm going to talk about some of the other interesting genes that we don't have much function for. The largest, the largest gene cluster we see have 585 genes, and it's annotated as a quinoprotein alcohol dehydrogenase that uses a pyrrole quinoline quinione or PQQ as a cofactor. These proteins are quite common in bacteria, especially in proteobacteria, but they're not very common in archaea. In archaea, you can see them in these Bathyracota, and in halophiles, but other than that, they're not very common. So, and not only does the largest gene cluster have this annotation, there's 76 gene clusters in this pangenome with that annotation. So in this pangenome plot here, I've sorted the genes according to their presence-absent patterns. So all the genes in this part here are shared by many genomes, and then this part here is this variable gene, variable part of the pangenome. And the proteins or gene clusters with the PQQ domain is indicated with this PQQ bluish green. So you can see that there's a lot of these. The majority of them are in this variable part of the pangenome. And yeah, so this heat map here just shows the distribution of these, of the 12 top families in our MAGs. So each of the MAG has 4 to 64 genes with this annotation. And so again, here, each of the columns is a MAG, and each of the lines here is a gene cluster that has that annotation. So one interesting observation is that most of the MAGs have many copies of one of the gene clusters. So they don't have like one gene from each of the different gene clusters. So this suggests that this kind of expansion of these genes is kind of a recent event, and it's probably an ongoing process. So for instance, all of these 6A ones have a lot of these genes from gene cluster 1, while the ones from the 6B clade has a lot of genes from the gene cluster 10 here. instead of having several of them from several different groups. Okay, so what do we think these genes are doing? So when we look at the domain structure from those different families, they all have they look very similar. They have like an extracellular portion, and they're very similar to methanol and ethanol dehydrogenases from bacteria. Or not very similar, they're similar. When you look at the phylogenetic tree, these are the bacteria here, and these are the Brachyarchaeota families that we have. So they're quite distantly related, but they are they have become the same structure. In bacteria, these form these extracellular dehydrogenases that where the PQQ cofactor feeds electrons directly into the electron transport chain. So it makes it possible for them to kind of get energy directly from oxidation reactions. So that could be what they're doing in these Brachyarchaeota too. Okay, so another very common domain that we see in the pangenome is these B12 radical SAM methyl transferase gene clusters. So I've indicated those here in red in the pangenome plot. So here we also see them both in the variable and in the kind of the shared part of the pangenome. But for these ones, the majority is found in the shared part. And there's 25 of these radical SAM methyl transferases in the pangenome. This here just shows a phylogenetic tree of these gene clusters. So the closed mag has, I think it's 17, it says up there. They have 17 of these genes. And so the expansion of these one is not as huge as what we saw for the PQQ genes, but still, like, it's a huge diversity of them. So it suggests that they probably are important too for their metabolism. Okay. Yeah, so what do we think these radical SAM methyl transferases do? So as I said in the start, like lots of the studies of these MAGs have suggested that they use methyl or methoxy groups from lignin for cytogenesis. In the termite paper I pointed out, they suggest that they use these two genes here. So this is a tetrahydromethanopitrine methyl transferase that then transfers the methyl group from their methanogenic corinode protein into the methyl branch of the Wood-Longdale pathway. And we do find both of these two genes in all our MAGs. They're always found together in the MAGs that have them. And they're always found with a third protein that is just annotated as DUF1616. So this is a small membrane domain protein, so maybe it just anchors it to other proteins to the membrane. But these kind of systems also need a substrate-specific methyl transferase. Okay, so we think that this is probably where these B12 methyl transferases come in. Okay, so we also have some other possible lignin or lignin cellulose genes. So as I said, the MCG8 genomes have this UBIX, UBID system. For anaerobic benzene degradation, you need both of these genes. So we can only find both in the MCG8 group. So we think these might be used, or this group might be able to do carboxylation of aromatic compounds for degradation. We also find these other genes here in some of the MAGs. All of these do need oxygen or hydrogen peroxide to work. So it's unclear, how much oxygen these organisms see in these systems, at least, maybe when they live in a deep subsurface. We also looked for K-zymes to see if they had other enzymes that are kind of active on plant materials. We did find that all of the MCG6 MAGs tend to have these G5 enzymes, which could be cellular, work on cellulose or mannase. since we did not see them growing in the cellulose cultures, we think that these probably are manaces. They also have genes for starch and glycogen degradation. And we see more of these genes in the MCG6 genomes than in MCG8, which probably explains why these are the most common ones in the bioreactors and the plant cultures that we have. Okay, so this is extracellular peptidases. We also see those in the culture in the MAGs, suggesting that they could break down proteins from the plant material. And again, we see more in MCG6 than we see in the MCG8 genomes. So to sum up, we think that these protearchaea can be involved both in the hydrolysis of the complex compounds in the plant material, but they're also probably involved in this cytogenesis. And the reason that they are always positively correlated with the hydrogenotrophic methanogenesis and metallotrophic methanogenesis, probably because they like the same conditions. They will also contribute to their community by doing nitrogen fixation and by producing vitamin B12, which is a good commodity for communities. And from an evolutionary point of view, I think those gene expansions that we see are very interesting. So we're going to look more at those. We also get some clues from the MAGs how to get better enrichment of these MAGs. For instance, we can add this PQQ factor, which when you grow E. coli, it will grow better. And maybe not having any nitrogen source would help us enrich for the MCG6 genomes. For MCG8, they don't have the B12 synthesis pathway. You could probably add vitamin B12 and get more of them. So the next steps here would be to get a better protearchaea culture and then do some proteomics. I had hoped to have some proteomics ready for this, but that didn't work. So, yeah, so then thanks to all the people who are involved in this, Ivy, Thorstein, Mabel made the cultures, and Upama here, she has maintained those cultures, which is great. She's going to talk to them about them too. And Elizabeth and Amar, PIs at Bioson. Ilya is, unfortunately, he's stuck in Russia. He's actively seeking work outside. So if anybody knows, he's the expert on metabolism and external questions.
Thank you very much for this, It's on? Okay, very good. Thanks a lot, Camilla, for this insight into this interesting group of archaea. Is there any questions from the audience?
Hey, no, I'm just a bit curious about the gene cluster that you mentioned on slide 14.
Yeah.
So you said that it's the largest gene cluster.
Slide 14.
But,
the largest, yeah, for the PQQ one.
yeah.
Yeah.
You know, like,
yeah.
Why is it so large?
Yes, why are they so large? That's a very good question. And I think also one of the reasons that they're so hard to assemble is that they have these lots of these repeats in their genome that are very similar. So, you know, to me, it kind of suggests that maybe they are important in their, they must be important for something in their genome. Look, now it's gone. You can see the top of my yeah, so they can have, the closed MAG has 44 of those genes in the genome. And then they can have up to 64 of those genes, and they are they're always similar within the genome, but they look to be diverging quite rapidly. We did try to do AlphaFold on them, and AlphaFold could not fold the whole protein. So we have to kind of cut it up into smaller pieces and fold different parts of it. Yeah. Mm-hmm.
Yeah, trouble.
their treatment.
Is it possible to do a rough estimate of the amount of worldwide nitrogen fixation that these might be responsible for? Based on
Yeah,
how much,
yeah,
what the distribution is of the NIF genes?
yeah, that would be very interesting to do. It's only, the NIF gene seems to be also in those MCG6 genomes and not in the MCG8 one. So yes, that would be, they probably, if, they say that the MCG6 are the most common on the planet, they must be contributing a lot. But I don't think the NIF genes haven't been assigned to those genomes before, so I don't think anybody has actually looked at that, so that would be interesting to look at. Yeah. Mhm.
Yeah, Gabrielle.
Thank you. Thank you for your talk. I have a question regarding the genes that are putatively involved in lignin degradation or modification.
Yeah,
You did not find any lactase or
No.
nothing,
No.
and peroxidase.
yes, we did.
Yes, the
Yeah.
last ones.
Yeah, yeah.
Okay. Okay, and
We did find peroxidase, but it wasn't, it looks more like one of those peroxidases that are just involved in just oxidant kind of detoxification, not, they didn't cluster nicely with any of the ones in the case
Okay.
database.
because these enzymes are pretty interesting for
Yes.
bioremediation. So
Yeah,
do
yeah.
you plan to subclone these genes to produce the enzymes?
Yes, I think what we will, we want to at least start out with kind of trying to get like a culture that has a lot of them, so that we can isolate proteins maybe and see what they are expressing at under different conditions.
Okay.
And with all these oxygen genes, and they are, kind of anaerobes, like what they must be getting oxygen at some point to keep these genes in there.
Yes, for sure.
Yeah, yeah.
Okay, thank you.
I was thinking about the same
Yeah,
things
yeah.
until I realized, okay, this is an anaerobic environment.
Yeah.
So in that case, it's like
Yes, I
using
think maybe
oxygen
in
as-
the bioreactors they might not do much, but maybe in the subsurface, because you can have small amounts of oxygen coming in, and
yeah, that's
yeah,
true. Yeah, absolutely. Oh yeah. Can we see the chat? Or you can maybe read it up if you see it. Okay, you can do it. That's good.
So Joe Handelsman says, great talk, Camilla. It's been suggested that the Bachyarchaeota evolved in a hot environment. Do your data support this?
if my data supported, I haven't looked at that, but that's, yes, I agree, and it would be very interesting to look at, because that's one of my other interests is the evolution of temperature, and I've looked at that in bacteria. So that's one of the things that I want to look at, like, do we see the same gene, like in the bacteria I looked at, you can see that they kind of have imported lots of genes from low temperature bacteria, for instance. So I think many of these kind of novel genes that we see in these archaea, so the nitrogenase, for instance, and also those PQQ genes, of course, like they're only found in bacteria and halophiles. I think, you know, one of the things they say about archaea is that they take in a lateral gene transfer and then they expand them and use them for different things. I think that's probably one of the things that we see here is like they come into a new environment and they get these new genes for living at lower temperatures. Thank you.
Okay, thanks a lot again, Camilla. Okay, now we come to the final talk, this first session, which will be by Anupama Sharan, also from the University of Toronto, Canada. And the title of her talk will be Developing an in silico pipeline for the mining of anaerobic O-demethylase enzymes from lignocellulose-rich metagenose. Anupama.
So I can control the screen? Yeah, I'm on the Zoom call. Yes,
Let's work out.
I figured it would be less clunky. Yeah, rejoining as a panel. Okay, awesome. Yeah. Okay. Thank you. It looks good. So yeah, good morning, everybody. Thank you for this opportunity to give a short oral talk on the poster that I'm presenting. And anything I cannot cover in this short 10 minutes time, please feel free to come check out my poster, and I would love to talk to get some more ideas. So I am a part of the same group as Camilla at the University of Toronto. And yeah, sorry. Thank you. And my project is also a part of this bigger Canada-wide collaborative project called Synbiomics, where we are very interested in looking for new enzymes that will help us functionalize different parts of biomass. And for my talk today, I'm focusing on a group of enzymes called O-demethylases. And we are very interested in seeing if these enzymes can work on aromatics or oligomeric compounds that result from lignins specifically in the biomass. And I'm a PhD student, and I'm supervised both by Professors Emma Master and Elizabeth Edwards, and Camilla is also collaborating with me on this project. So just a quick introduction to the biochemistry. The O-demethylase systems, these are coronoid-dependent enzymes that are found in mostly acetogens and organohalide-respiring bacteria. And what the system does is demethylate aromatic compounds. These aromatic compounds are actually formed by ligninolytic enzymes, so lactases. There was a question earlier about that. So fungi release these in nature, and then they become available to the soil microbial communities. And once in the soil or any other anaerobic subsurface environment, the bacteria work on them and demethylate them to, And yeah, so this function is really key to increasing the reactivity. And the methyl group, which gets transferred over to the second methyl transferase, MT2, then goes on as it helps in energy conservation in these microorganisms. And this coronoid-dependent protein, this acts as a methyl shuttle, and it shifts between a reactive state when it's accepting the methyl group, then it becomes very reduced and inactive once it transfers the methyl group over to the second methyl transferase, and it is regenerated by a fourth enzyme, which is called activating enzyme. It's not on the slide here, but the reason it's not here is because I wanted to show that these three enzymes, they are co-localized, so they occur together in the genome, and the fourth enzyme occurs elsewhere. And this co-localization feature becomes a very good and helpful mining feature when we want to look for these systems in metagenomes. So I'm using this co-localization feature to mine these systems from different metagenomes. And once we have the more reactive components, these demethylated aromatics, they are very useful for the pharmaceutical industry and aromatics and fragrance industry. So there's a number of applications which you can do, and these become platform products then. So the specific research objectives, this is actually, in fact, going back from function to sequence. So we have a function in mind. And then I wanted to see, well, how did these enzyme systems evolve? What does the ecological distribution look like? So the first tool I developed an in silico mining open source platform. So this tool lets you look for these systems in your own metagenomic datasets. And I use this tool specifically in environmental datasets that I know are rich in lignin and aromatics. And then finally, I was fortunate to have some of the active cultures growing, so I could do some proteomics experiment and also confirm the expression of these systems. So this really is the paradigm going from sequence to function, or really function to sequence, and vice versa. So the first objective, developing a open source pipeline to mine these co-localized OD methylase sequences from metagenomes of interest. And we start by creating hidden Markov model profiles. But the key here really is to use biochemically characterized sequences. We don't want to start without having any evidence of function, but we want to incorporate that functional evidence in the tool itself so that your models are developed like that. And the sequences that you get back, there will be a higher probability that these are functional and these are expressed. And the other steps in the pipeline, they are pretty straightforward. I know there are a lot of experienced bioinformaticians in the audience, and this might be very simplistic, but trust me, when I was starting out, there was a lot of hair pulling going on because the information was not very organized. And this is a very desirable organic chemical modification. But if we want to go back and look at public databases, the information is not very organized. So I made this tool out of frustration because there was not a lot of modular or tutorial style tools out there that would let you search your own data set for a specific function. So you can use this tool if you want to look at the same enzyme systems, but the co-localization algorithm in this tool lets you do that for basically any other system which might have the same marker. So you can use the tool as it is, customize each module, or you can also treat it as a suggested workflow and apply it to your own project. And then finally, once we get the sequences back, you have the option to visualize it, either in the form of network diagrams or the more traditional phylogenetic trees. And in the pipeline that I have on GitHub, there are R-based tools for the visualizations. But the results that I'm showing today, I have used a slightly different tool, which is cited here. That's also open source. And it helps you make network diagrams. And then I've styled them in Cytoscape. So the first result from the in silico mining. So what I did here was I had the metagenomic data sets I was interested in, and there were around 13 data sets that I looked at. All of these data sets, which you can see in the legend, they are available on JGI IMG. And I made these network diagrams. So each node represents a sequence that I obtained using the HMM search tool. And they are colored by the metagenome from which they were obtained. So this is for MT1, which is the environmental substrate interacting methyltransferase. And this is the clustering that happens. So clustering here is by sequence similarity. And you can see at the bottom that it is quite a high cutoff for sequence similarity. So these sequences are very similar to each other, and that is what the edges represent. But we also see that there are sequences which form these orphan nodes, which don't really cluster with the others. And they are very interesting novel candidates because they still come from a search using biochemically characterized sequences. And then I did the same for the other two enzymes in the system. And also each node here is co-localized. So for every MT1 node, you have a CP node and you also have an MT2 node. So this is exclusively with the co-localized enzyme systems. So what was really interesting here in these diagrams is that you see that the colors are quite dispersed. So all these systems, I thought, well, and I think Dr. Alam said that there's not a lot of genes that are common to different microbiomes or metagenomes. So I thought, well, if there are different aromatic substrates in each of these metagenomes, which could be directing sequence evolution, then you would actually expect that the clusters would be, the colors would come together in the clusters if each metagenome had its own unique OD methylase system. But instead, what we see is that the colors are quite dispersed throughout. And I unfortunately don't have 16S data, but from the MAGs or the binning data, I know that this clustering actually happens by taxonomy. So this tells me that the O-demethylase function is actually encoded by a limited number of taxonomically diverse organisms that are common across all these different metagenomes. And another very interesting thing, or well, the hypothesis that was, the hypothesis that was, I think, proven by this analysis is that the percentage pairwise identity for MT1 is the smallest and MT2 is the highest. So MT2 is the most conserved. MT1 seems to be the least conserved, and that is supported by their biological function because MT1 is the environmental substrate interacting methyltransferase. So we would not expect that to be very conserved across different environments, whereas MT2, which is the metabolite interacting methyltransferase, that must be conserved because those metabolites are common across all these organisms. And that is also supported by this sequence analysis. So I had access to, fortunately, those metagenomes, which were microcosms started by Mabel, and I was able to do some proteomics on it. And I sampled these microcosms, which were enriched on the polar hydrolysate that Camilla presented on, and also cellulose. And the hypothesis I wanted to test here was if the O-demethylases would be expressed more in the lignin-containing substrate rather than the cellulose substrate. So that is what I wanted to check using proteomics. But one caveat also of proteomics is that once you get the data back, it depends a lot on the database from which you're identifying the proteins, because it is peptide matching to whole protein sequences. So depending on the kind of database you use, you have to be careful because initially I put in all the coding sequences for the metagenomes and I did not get any hits back to my O-demethylases, and I was very disappointed. But then when I cut down the data set to the smaller ones exclusively for O-demethylases, then I got a lot of hits back because the software cannot distinguish between homologs. So for anybody who's planning proteomics experiment, please keep this in mind. And then the last result, the same diagrams, but now they contain the expressed candidates. So none of the expressed candidates came from the co-localized systems that I showed before. So I had to manually add them into this network diagram. And we can see that here, the sequence similarity is very low. And I expected that because they did not come from the co-localized sequences. And when we also add them, even at this low similarity, we see that they cluster quite differently. And what is interesting is that I found more O-demethylases in the cellulose microcosms rather than the polar hydrolysate. And it was a little disappointing. But I also know that these microcosms have the same organism. So the same organism, even if it's present in a cellulose enriched substrate, would still be encoding the O-demethylases. And we could argue that the cellulose enrichments are less stressed than the lignin containing ones. So they are healthier and you would capture more proteins from them. So it's a matter of like sampling and how healthy your cultures are. So quick conclusions, I found 25 novel co-localized O-demethylase gene systems in around 13 data sets that I looked at. And the proteomic studies confirm the expression of those systems. And as we saw from the diagram before, limited number of taxonomically diverse organisms encode them across very different environments. And a very important thing to emerge out of this was that when looking at metagenomic data, we think that the more stringent we are with sequence features, we have a better shot at capturing genes that would be good for heterologous production, but that might not necessarily be true. So it is very important to look at both the express data as well as the sequence data and then select candidates for heterologous production. And future direction, there's definitely the need for a functional assay to screen this O-demethylase function in metagenomes. And then we can truly bridge this gap between discovery and function or sequence and function. And I did some initial work with an undergrad student on the functional assay. Unfortunately, it did not work out in time. His thesis finished. So if other people have ideas about anaerobic enzyme functional assays, I would love to hear them. So yeah, I would like to thank my supervisors, Camilla, for doing a lot of help with the bioinformatics support, and Robert Flick, who helped me with the proteomics work at Biozone. Thank you.
Thanks a lot, Anupama, for this nice work about the methodaces. Okay, one quick question by Sean.
EST.
The networks.
Yeah, the sequence memory network.
Yeah, no, that was locally. It was using that tool called SSN5. So I just had it downloaded to my system and I made them locally.
Right. And how do you decide which e-value you choose? Because I think depending on the e-value, your network can change
Yeah.
a lot. So,
I mean, I think this was more for aesthetics rather than knowledge, because at a I wanted to choose the highest cutoff at which there was still good clustering that I could see, because it is not very informative. If I chose a cutoff higher than this, then there would be a lot of orphan nodes that get thrown out. But because the diagrams also contain the nodes from public databases such as RefSeq, I wanted to choose like a sweet spot where I could see clustering with the public databases and also infer some more information like that. So it was, I would say, like manually the choice of e-value was done.
Okay, thanks a lot, Anupama, and thanks a lot to all speakers of this first session, which I herewith close. And we will now proceed to lunch and reconvene for session 2 at, what was it, 1:15.